friends, good morning, namaskar. Welcome back to our second lecture of zoology introductory session. From the next lecture, we are starting with actually syllabus of zoology. First lecture will be on cell biology. But before that, this is a short video which is giving you information about how our YouTube lecture series will be and some information on the book. Some of you who might have not purchased the book, I will suggest you to purchase book. The link will be posted in the description. Also, join and subscribe to my YouTube channel, and a link to Telegram channel will be also provided in the book. Okay, so let's start with our uh, first slide. So this is the study plan of how our lectures will go. Right. The overall, I have classified made a pattern of all the zoology topics in the zoology syllabus. Okay, you might see that they are not according to the syllabus as given in the UPSC pattern. Right, they have started with the non-coordinates and non-coordinates, and then ecology, ethology, all that. But what I suggest is that for anyone who have any zoology background or biology background or no background, okay, for them it is better to start with the basic cell biology, right? So that's why I suggest you to go with this pattern. Like, first start with the cell biology, then biochemistry, genetics, and development biology, systematics. After that, this is what contains the basics of zoology and biology, right? Once you have completed with this, you can go for the further study in it. Then you can start with evolution, physiology of humans, non-chordates, chordates, ethology, ecology. All these are kind of applications part of the biology. Okay? So you should go by this pattern of study. That is going to be helpful. Most of the time, what happens is that as the chordates and non-chordates are the first chapter in the biology, some students try to start it, but trust me, chordates and non-chordates are the most difficult part of entire zoology syllabus. Right? The first reason being that the notes available are not precise; they are so bulky. So students get frustrated when they see a 3 or 4 pages classification in RL code file or even other evolution notes also. So that's why we'll start with the non-coordinates and coordinates. We are going to conduct lectures, and we are also going to have notes on it, which will be very simplified. Right? So don't take tensions of coordinates and non-coordinates. So first, tomorrow's lecture will be start with the cell biology, and this is the entire syllabus. Right? So I have enlisted all of these topics and subtopics in it. Along with that, some additional points which are not there in the syllabus, we have to complete them. Yeah. And how did I find these topics? Is from the analysis of the question papers of 22 years, right? So that's why I was suggesting that even you should go for that question paper book. So that is why it's more important. And it's not about what money I make from that book because it's only 120, and if you know, after getting it from Amazon, I get a little share about that 120. I don't get the 120 out of 120 rupees, but the book is really helpful. It has helped me a lot to analyze the subjects because most of the time, because the syllabus is so bulky, we don't get any direction, and even if we study, something different than what is going to be asked in the exam. We are going to analyze the actual examples of it. Right? So just wait for it. So after this third biology, this introductory chapter, we are going to get this one by one. And if you look at these points, some points are like that we can cover in less than and half of the lecture. Some points are there we may take more than one lecture. So don't consider like that all of the points we will cover in the one lecture only. These are just classification which I have given, and not the separation of the syllabus, right? Okay. Next is the how to study any better topic. The first is the hand drawn diagram. One of the most difficult part is drawing a diagram. And when you see a diagrams in a book and don't draw it on your own, it becomes more difficult. Right? So if you have hand-drawn diagrams of anyone, you will also learn how he has drawn. I'm going to draw those diagrams on the LCD screen. So that's why you are going to learn how to draw it perfectly and in an easy way. Okay, so that is the biggest help I'm going to do provide on this YouTube channel. A complete set of diagrams, right? Then also flowcharts if there are any, theory part also, and the tables including. All of this we are going to uh, a theory part and all the result notes. Actually, I have not planned it well, so maybe those notes may be available either on Amazon Kindle only, or you will order from directly from me, and I will send you a print PDF, a print digital print book of the notes. Okay, so that's we are going to decide when we complete all the points in cell biology chapters. Okay. After that, more gates will also open after the May. We pray that the lockdown may get over, so that's why there won't be a problem in delivering goods and the books also. Right, so this will be the pattern of our study, and I'm going to solve your biggest problem of diagram. How you can make it simple. Okay. Next, now I'm saying you that the good is book, but does it really helping? So I'm going to give an 3 to 4 example of cell biology. Okay, so I have displayed here some few questions of cell biology. Okay, I'm not going to display further questions of the book because this might be a those who have purchased books they may say that if you are showing all the books on the YouTube, why you have asked us to purchase it. Okay, so I'm just for just as a sample, I'm just showing four pages of that book here, and let's see how the book is really helping. Okay. So first of all, this is the syllabus point given. Okay. Structure and function, right? Structure and function of cell. In that they have given nucleus, plasma membrane, mitochondria, Golgi bodies, endoplasmic reticulum, ribosome, lysosome, and others. Now, what we expect the questions the UPSC might ask are there. Those questions should be based on only structures of how cell is, right? We just expect the questions on those point only, right? We expect the questions on those point only. Okay, but that is not the case. Here, what we want to make a difference from other students preparing for this exam, right? How? Let's start with the analysis. The first question I asked in 1998, what was almost marvelous. Distinguish between cisternae of Golgi complex and that of endoplasmic. Right? Even if the analyze, you have to analyze that your work is not only to study the individual parts of the cell, like the mitochondria and endoplasmic reticulum, rough endoplasmic reticulum, right? And then Golgi body. You are not Your work is not just up to the part that you are going to study its structure and what it does, what it secretes. Right? That is not only your function. That is what I want to convey to you. That is how we are going to dissect the syllabus so that we won't miss a single question and we won't be surprised when the questions will come in the exam. Right? We are going to minus that, eliminate the surprise factor from the question paper. Right? So you are what I'm going to I want to say is that you will also need to compare these organelles with each other. If you have been asked that the right structure and function of mitochondria, you will write it. You will write a 3 to 4 pages even. Right? But when you will be asked to compare or the distinguish between mitochondria and Golgi bodies, you should know what are the distinguishing factors and what are the points which come in opposite or in similar way. Okay, so that is a comparative study is required among all these organelles of the cell. Okay, so that should be your approach. This is an example of it that they have asked about the difference in between. Next is the distinction between flavoproteins and cytochromes. Remember, these are not mentioned in syllabus. This is not the case. These are the subparts of the organelles. These are inside the part. So they have not asked only the distinction between nucleus and endoplasmic reticulum, mitochondria, and RBC. So they have gone ahead of it, right? So a note on lysosome is the comparatively easy. Now this is the questions where the book will play, the analysis of the question papers will play here. What is the question? Mechanism of elaboration of secretory and plasma membrane glycoproteins. Okay. You know there are glycoproteins are three types: secretory, plasma, and lysosome. Right. If you're looking at the syllabus point of structure and function, right, you might do the structure and function of plasma membrane, lysosomes, and even function also. But what has been asked is about the mechanism of it, how it releases, right, those glycoproteins. That is the part you might have thought that this is not the part of our syllabus. What is the part of our syllabus? Structure and function. Mechanism is not, right? But this is not the case. They have asked this question in 1998. Okay, so this is the first case of how the technical analysis is going to help us to change the way we study the zoology. The next is the function of Golgi complex. Easy. Rough endoplasmic reticulum and smooth. Easy. Distinction between lysosome and Golgi bodies. This is also easy. Describe the structure and function of ribosomes. This is also easy. Next is what? These are all the easy questions. The structure and function of endoplasmic reticulum. Explain the structure and function of plasma, lysosomes, distinction between peroxisome and diaxisome, short note on chloride shift. So, this may come under cardiovascular system also, but it happens with the RBC, so that's why it may also come in this point also. So don't say that this is not the question I have this topic. And this is the structure of mitochondria, plasma membrane, endoplasmic reticulum. Same structure and functions. Okay, so this is the question. If you have not studied the question paper, this you will say that only this kind of question will ask. This is the structure and function of plasma and endoplasmic reticulum, structure and function of nucleus, structure and function of Golgi. You will say that only this question will be asked. Okay, but this is only one question directly from you can say directly asked from the point of the syllabus. Next is the difference between Chantal-Garneau sequence and Greenbow sequence. Okay, this part also contains the transcription and all that. Okay, but the Chantal-Garneau sequence is the ribosomal binding site in bacteria. So that's why in the structure ribosomes it may also go. So that's why I have just classified under this chapter. It may also go transcription also. Next is the what are suicide bags of cells. Briefly describe the function, lysosomes, and all. Next is what. So this is what. After reading this, you will get to know that you will also need to go through all the or names of organelles. What are the suicide bags? Right? What are the unnatural? Sorry, something like that question was that organelle in the cell. Okay, so that's part of all the or names or the second names of the organelles you need to cover. Right. Next is the this is the important point. Second, first one was the earlier. This is the second. Now look at this question. Which organelle is well developed in a secretory cell? Describe the structure to justify its function. Right. Now this is the questions which requires a highly in-depth knowledge and study of zoology. Right. After reading these kind of questions, it tells us that we are not going to get a superficial questions in the zoology UPSC paper. We are also going to get some in-depth questions where we where we really require to study zoology. Right? So this organelles, well developed and secret cells. This kind of terminology you will get only when you have an entire and thorough knowledge of the books, right? Whether it is Golgi bodies or endoplasmic reticulum. Right? This is the questions. Most of you might not even know that uh, there is some kind of thing that so, which is the organelle developing secretory cells. Many of you might not understand the nature of the question or the what is this question about. Right? So that's why we require to analyze this question paper. And when we will study this cell biology chapter from our next lecture, you will going to understand such terminology also. Right? So next question. These are all almost easy. 30 years and this difference. What is the difference? This also can comes in transcription and DNA technologies. Mention the role of ion channels. Okay. Describe the organization prokaryotic cell. What are the traits and functions in which it differs from eukaryotic cell? Okay. This is the basic terminology question. Difference between eukaryote and prokaryote. Right? This is the F1, F0 partition mitochondria. This is also easy. Most of the time you will study about these also. List and illustrate the components of biological membranes in terms of arrangement. Next is the univator, endoplasmic reticulum, smooth and rough. This is the structure of function of lysosome, a straightforward question. Explain detailed structure, organization and functions of Golgi, a straightforward question. Now, these years they have asked a straightforward question, but in 2018 they have asked a question. Explain the origin and functions of lysosomes, right? With emphasis on different enzymes, right? Whenever we study a lysosome, right, we are not going. We usually don't study details the lysosomal enzymes. Right? We study the roles of lysosomes only, but not the enzymes. So this is what I have observed one time. But from this question, you will understand that the depth of the questions is not superficial. Right? Under this question, when you are going to study lysosome, when we are going to study cell biology, we are going to study, may give to enough time for each and every organelle that we won't miss such a simplest or a minute point from the cells. For some, it might be a hectic job, right? But I'm going to do this hectic job for you for once. So when we complete this entire lecture series on YouTube, a time will come that we will say that even if any questions will be asked from zoology in this question paper, we will able to solve it because we would have already covered everything about that. And some students say that don't go in depth of every chapter, just go for the previous year questions and only just cover 80 to 60% of syllabus. It would be fine, but it's a guarantee that we will score 100, 250 plus, right? But my job is to give you 100%. Out of that, you can choose which topics you have to cover and which topics you don't want to cover, right? So these are all the coming back to our question. These are all the lysosomal enzymes, right? And you will fetch almost full marks if you enlist these types of lysosomal enzymes in your answer. Four types of enzymes are there. If you just enlist 5 types of enzymes are there, if you just enlist all of these enzymes along with their function, you will fetch full marks, right? Because question specifically talks about what with emphasis on different enzymes of the lysosomes, right? You have not been asked about the structure and functions of lysosome along with only the function of lysosomes. Okay? No, you have been asked the questions related with enzymes. So you have to be specific where from these enzymes come, where did they act, and how did they act? What are their functions? That should be your emphasis. And once you have gone through this question, you will understand. Yes, in-depth analysis, in-depth knowledge will be required. Otherwise, you अगर आपने क्या किया होता if we have done all the topic of the cell biology, all the organelles, but only with the structure and function, you will not be able to answer this question even after doing that, right? So the in-depth knowledge is required, and we are going to provide you. Also, you should also try by referring books, right? So next questions are other questions are easy, right? Uh, is a fluid mosaic model, electron transport system in mitochondria. The second question, the fourth question is that they explain the isolation and methodology of study of the cell membrane. Right? Now, this question has been asked in the last year. Most of the time, when we study the cell membrane, we study how the different models of cell membrane are, how the structure is, right? What are its function? We do the detailed analysis, right? 5 to 4 page notes. Right. What has asked in the last year? They explain the isolation and methodology of study of cell membrane. See the question from out of this world. Sometimes I think, right? No one asked this question yet. Why would you ask the methodology to study the cell membrane if only the structure and function is given in the syllabus? But we cannot challenge UPSC, right? If the questions are coming like that, we will have to change the pattern of our study a little bit, right? And I suggest you don't take tensions about what points are necessary and what are not. I'm going to do that hard work for you. My aim is to take what I'm thinking about zoology as optional is that in 2008 all India rank 1 was having zoology as optional, but after that, because of the scarcity of the material and the good online courses or even offline courses, the students giving zoology have been decreasing, right? But I want to make a small effort in taking zoology to a higher level to get more students taking zoology as an option. And I think I can contribute a little bit. So this is the questions which I wanted to share with you, right? So that is why I suggest you to go buy that book so that you will understand what kind of study. Now after going through these questions, you will look with a different eye to that syllabus. You will look with a different perspective to the syllabus. Right? While reading, you will not just read structure and function of your draw lysosome. What are the structure? These are the part. These are the 10 functions. All that. You will not do that. You will try to compare it with others. You will also try to go in depth analysis of it. Right. So this is up to you also. If you find it useful, just purchase that book and increase your Kickstarter study. Right. So now I have announced in the Telegram group that those who show the trust in me by purchasing the book, I am bound to give them something more. Right. So for them, I will form a separate Telegram group so where I will be posting more frequent updates. Right? It's not like that I'm just going to neglect the current book. It is will remain on the highest priority. But I'm going to give. See, this is the mostly one slide. That this is the concept now. I am starting from today only. I'm going to give one slide or note on each many important topics. Right? In that one slide, you will have a diagram, notes, function, and all that is required in a short. Right? So the mostly one slide will be more frequent in that group because they are the most serious. They might be the students who are actually giving mains this year, who have actually qualified last year. Okay, so I will be also in regular touch with the regular group. So anyway, someone might say that now you are going to be so being commercial. This is not going commercial. This is I'm doing because I want inspiration or something a drive to do all this work. Okay, because I have 4,500 members in channel, 1,500 members in groups. But I know there are very few students who actually inspire me to do various things. Right? So I want those serious candidates who will actually keep me motivating. Okay, motivation I was to talk about. they will keep to motivate me doing more and more things. Okay, so whenever you are starting something free on anywhere, you need motivation. So for that, I'm going to form a separate Telegram group. Okay, and also I will get their queries answered on live sessions of YouTube. Meanwhile, every 8, 10 days or even 7 days possible. Then if there are any products coming, like the notes of cell biology might be the next product. So I'm going to give a discount on that. And how you can convey me that you have purchased my book, that I request you to send your private message me on Telegram your Kindle ID and a screenshot of your Kindle store, which you will show my book on it. Right. So I will make sure that in the your Kindle store ID will not be misused by me or anyone else. Okay, so this is the what I have to say about how our lecture series will be, how the book is going to be, and our next lecture will be on cell biology, the first part, maybe if possible today evening or tomorrow. Okay, have a nice day. Goodbye.